Each cell in the human body is wrapped in a membrane that separates the inner environment from the outer environment. And positively and negatively charged ions aren't equally distributed on both sides of the membrane. Fundamentally, it's these differences in concentrations and charge, as well as permeability across the membrane, that establishes the cell's resting membrane potential. Generally speaking, there's higher concentration of Na plus or sodium, Cl minus or chloride, and Ca2 plus or calcium on the outside of the cell, and a higher concentration of K plus or potassium and A minus, which is just what we write for negatively charged anions on the inside of the cell. These anions include a variety of amino acids and proteins that are produced by the cell. Let's start with the sodium potassium pump, which uses ATP to move 3 sodium ions out of the cell for every 2 potassium ions that it moves into the cell. This is like the workhorse of the cell, and it helps establish the concentration gradient for potassium and sodium. So let's focus on potassium, which has a concentration of 150 millimoles per liter on the inside of the cell and about 5 millimoles per liter on the outside of the cell. With so much potassium within the cell relative to outside the cell, there'll be a fairly strong concentration gradient moving potassium ions out of the cell. Although these ions can't simply diffuse through the phospholipid bilayer membrane, it turns out that potassium can get across the membrane using potassium leak channels and inward rectifier channels that are scattered throughout the membrane. So using those channels, the concentration gradient pushes potassium out of the cell, and that potassium brings with it some positive charge and leaves behind some unpaired anions, which carry negative charge because they aren't able to go through the leak channels. Over time, as more potassium ions leave the cell, a negative charge builds up within the cell, and this starts to attract positively charged potassium ions back into the cell. And this is called the electrostatic gradient. This electrostatic gradient is established with the movement of relatively few ions, so it doesn't upset the overall concentration gradient that was already established. For potassium, the exact point when the potassium moving out of the cell due to the concentration gradient equals the potassium moving back into the cell due to the electrostatic gradient is called the equilibrium potential, or Nernst potential for potassium, and it's about negative 92 millivolts.